should I use, Corey?
Blue. Oh, wait, yeah.
There's choices.
Blue, rainbow.
Oh, okay, right? Sure.
All right, we have somebody drawing. Anyone else want to join her?
I don't want to be wrong.
It's okay to be wrong. That's what we learn.
Wait, hey Corey, you want to put my lab binder out of my backpack?
No, you're supposed to do it from the
Okay. Is this one or two pairs?
2n equals 6.
Oh, it looks like a little bit.
Anyone else want to come up and draw a stage? Get to use fancy, colorful chalk. Do you wanna? I'm not gonna call on anybody because I know it can be intimidating.
What is this, Medphase 2?
Metaphase 2. All right, Allison's going for it. All right, we have these daughter cells too. Anyone want to draw the resulting daughter cells? I'll stand up here and watch Allison. I'll turn the spotlight. All right, as Allison is drawing away, any questions about meiosis? I know probably most of you have gone through lab at this point. If you haven't, you're gonna dive deep into meiosis in lab. Yeah.
So like in meiosis 1 when it duplicates, is it a 4n subset?
Repeat the question.
So in meiosis 1 when it duplicates, like in the beginning, goes doubles and
Yes.
okay, so that's considered 4n subset.
So I would say 2n. Will you just tell me? Yes. So I would say 6. What is that?
Is that my alarm?
Yes, but there's 3 sets of 2. Yeah, so 6 total divided by 2. Yeah. Does that answer your question? All right. Any other questions about meiosis? All right, was anybody not here on Monday that needs this sheet? I don't know if we had few people this from sports and other reasons. The sheet basically saying if you wanted to buy one, probably install one. No worries. It might not seem like it at first. You will make it work. All right, so hashtag nailed it here. Thank you, Allison. Thank you, Rachel. So if we have 2n equals 6, what is the ploidy? 2n equals 6. What is the ploidy of this cell? Is it haploid? Is it diploid? Diploid. Yes. So it is diploid. 2n equals diploid. If I said n equals 6, then it would be haploid. Right? What does the 6 stand for?
Total number.
Total number of chromosomes. Yeah, so if we want to figure out how many chromosome pairs we have, we could take the total number divided by the ploidy.
Oh, that'll be 3, my bad.
So how many pairs should we have?
3.
3, yes.
I didn't see that.
That's okay. All right. So we should have 3. So we have some pairs going on here. I am going to draw another pair. I'm not changing colors, pardon me. All right. Okay,
Yeah.
metaphase comes after metaphase.
So
I'm sorry if that is confusing. Alright, so let's look at metaphase 1 first. Alright, so I'm just going to erase one of these pairs. Because we don't need one of those for 2n equals 6. Alright, so metaphase 1, we're lining up at the metaphase plate. Alright, this as a whole, what would that be called?
Tetrad.
Tetrad, also known as
Bivalent.
bivalent. Yes, I use bivalent in class. We use tetrad in lab. Alright, a single one of these. What is that called?
a sister chromatid.
Sister chromatid. You could also call it a chromosome. Yes, either would be correct. Alright, sister chromatid is what I'm going to label it. Alright, so hopefully everyone here has some version that looks pretty close to this for the metaphase 1 stage of our cell. Right, so if we go to anaphase 2, alright, at this point, right, we have our metaphase plate there. Our tetrads are separating out, right? So anaphase 1, tetrads are separating. We're now pulling individual sister chromatids to either pole. Alright, so that's our 2 stages in meiosis 1 that we're looking at. Let's look at 2 more stages in meiosis 2. So let's look at metaphase 2. Alright, if we look at this image, I'm going to draw in another set here. Right, is this drawn correctly? Yeah. So one of the major differences between metaphase 2 and metaphase 1 is that we only have a single row of sister chromatids instead of a double row of sister chromatids. Alright, so I know they're drawn in different styles here. If I were to draw it in that other style, well, just like that. So, single row of sister chromatids, double row of sister chromatids. So when we split out these sister chromatids, alright, we should get something that starts to look like that, right? We have individual chromatids being pulled apart. They are now known as a chromosome because they are on their own, right? And they're migrating to the pole. Now, of course, through all of this, all right, Allison has drawn it for us here, that we should have some crossing over. Because crossing over happened in prophase 1, right? So all of these will have crossing over having occurred. All right, so if we look at the resulting daughter cells, what needs to be added to this drawing in order to make it correct? Remember, daughter cells are haploid. All right, think about how many pairs we started out with to begin with. There should be 3, yes. They're not going to have pairs, all right, because they're not diploid anymore. They're only haploid. All right, so there should be 3. Each of them. I'm going to draw a shorter one and a longer one. Oh. All right. Any questions on what we just went over? All right, so we should be experts by now. If I ask you what are the total number of chromosomes in a cell that I have said 4n equals 8, what is the total number? 8. 8 is the total number. All right, how many different types of chromosomes? 4, yes. It has a ploidy level of 4. We could call that a tetrad, or not a tetrad, tetraploidy. Okay, I'm thinking back to my meiosis here. Tetraploidy. I don't expect you to remember that term, but that would be the technical term for this ploidy level. All right, and how many of each type are there? Yeah, 2. Exactly. All right. So we have, let's see, 1, 2, 3, 4. 1, 2, 3, 4. If we were to label them, I'd say this is type 1, this is type 2. All right. Any questions on sploidy? All right. Let's move on with the rest of this lecture. It's not too much more to cover. All right. So we looked at chromosomes. We looked at meiosis, mitosis. All right. But we can have variation within the chromosome structure and number. Right? Some of this variation is natural, some of it is not what it's supposed to be. All right, so variations in chromosome structure and number can have major effects on an organism if it's beyond what's expected. All right, so several human diseases are a result of this, It's important in the evolution of new species over many, many generations. We might get a new species out of this. All right, so chromosome variation, all right, on rare occasions, we can get the structure or number of chromosome changes so that an individual is different from other members of the same species. All right, they're abnormal. All right, between species, it's normal for a structure and number of chromosomes to vary. All right, so again, fruit flies, they only have 8 chromosomes. We have 23. That's a normal variation. Right, now if somebody had 24 and they were a human, that would be abnormal. All right, so we can have chromosomal mutations. All right, mutations within the chromosome itself. And we're just going to briefly go over this. If you take genetics in a few years, you will go over this into much more detail. All right, but we can have deletions. So we can have entire segments are just gone. So the segment E just been deleted. All right, we can have a mutation that results in duplication. Right, so instead of 1 D and E, we have 2 segments of D and E. We can also have inversions. Right, so the change in direction of the genetic material along the single chromosome. So instead of it reading DE, it now reads ED. All right, we're flip-flopping them. So think if we're flip-flopping them, this could have a major consequence in crossing over. Right, so if we want to cross over between the 2, D and E, we might end up with a chromosome that now has 2 Ds if crossing over had occurred. All right, so consequence of crossing over with these inversions. And then we have translocations, right, where one segment becomes attached to another chromosome. All right, so it can be a simple translocation where we have a single piece of a chromosome attached to another. All right, so these are not mutations within a homologous pair. These are totally different chromosomes coding for different type of genetic material. We can also have a reciprocal mutation, where 2 non-homologous chromosomes are going to exchange pieces, producing 2 chromosomes carrying translocations. Right, so now we have 2 chromosomes that are completely altered. All right, so there's these individual chromosome level mutations that can occur. All right, yes?
Would a reciprocal translocation be crossing over, like it's crossing over?
No, because it's happening on non-homologous chromosomes. So crossing over can only happen between homologous chromosomes. Yep. So in this case, it's not happening correctly. Maybe it didn't line up right in prophase, and that's how we're getting it. Maybe it is crossing over that's causing it, crossing over between non-homologous chromosomes. I'm actually not too versed in how this actually occurs, but I could see that as being one way that it could. Any other questions? All right, so that's change in chromosome level mutations. We can also have change in the chromosome number themselves. So if you're a euploid, that's the normal number of chromosomes. All right, a diploid organism, that would be 2 sets. That would be normal. We talked about a tetraploid organism. All right, that would be 4 sets of homologous chromosomes. All right, if you're polyploid, all right, that is when we, beyond haploid or diploid, that describes all other ploidy levels. So that would describe tetraploidy, that would describe triploids, 3N, and beyond. All right, so that's 3 or more sets of chromosomes. If you see polyploidy, that's what it's describing. We can also have individuals that exhibit aneuploidy. Right, so that's an abnormal number of a particular chromosome. Right, and this is actually fairly common to see. All right, so for example, if we have trisomy on 21, right, so chromosome 21, notice we have 3 homologous chromosomes instead of the normal 2. All right, that shouldn't happen. Something has occurred here, and when this happens, we get somebody with Down syndrome. All right, we have trisomy 13, also known as plateau syndrome. All right, in the 13th chromosome, we have 3 chromosomes. All right, shouldn't happen. These individuals rarely live past infancy. We also have the same thing happening in trisomy 16. This is typically pretty fatal and will result in a miscarriage. All right, so some of these mutations, these individuals can still live a relatively normal life, right? But some of these mutations are going to be lethal. We can also get mutations in our sex chromosomes, all right? So if we look at our sex chromosomes here, we should have an X and a Y. In this case, we have 2 X's and 1 Y, all right? This would be Klinefelter syndrome. This often occurs in males, and it's going to result in reduced testosterone, all right? If we look at Turner syndrome, that would be an X with no Y, right? This happens in females, results in slow growth and physical effects, all right? So we can have these changes in our sex chromosomes as well. Now, how does this actually come about? Yes?
It's a XO for Turner syndrome. Is the O just saying that there's no Y, or is there a chromosome?
The O is saying that there is no Y or an incomplete Y. Yeah. So there's 2 variations on that Turner syndrome.
Are we gonna memorize like different?
No, you do not have to memorize the different defects, but if I ask you, oh, can I have chromosomal changes in a sex chromosome? You should be able to say, yeah, because Down syndrome is one example of that. Yeah.
Wait, that's an example of sex chromosome?
Excuse me. No, totally wrong. I would say Turner syndrome is an example of that. In a normal chromosome, that would be an example of Down syndrome. Yes, good catch here. All right, so how do we get these abnormal chromosome numbers? This occurs through non-disjunction. All right, so during meiosis, we are not lining up and separating out our chromosomes correctly. So in meiosis 1, this can occur when we have one whole tetrad go to one side. Right? Meiosis 2, this can occur when we have a sister chromatid not splitting apart. Right? In either case, it's going to result with some abnormal chromosome numbers. All right, so this one has the normal number plus 1, same in this gamete, and these 2 are minus 1. All right, they don't have the normal number. They should have 2, but they don't. And in this case here, we have 2 that are perfectly normal. All right, 1 that's missing a chromosome pair, and 1 that has an extra. All right, so non-disjunction, that is how we get these abnormal chromosome numbers. All right, so if I asked you what process would create an individual that has Down syndrome, right? Non-disjunction. That would be the specific process by which that can occur. All right, so as I mentioned, this is typically lethal, all right? We really don't see a lot of people living past, infancy or even living to birth if they have abnormal chromosome numbers. We do have those instances where in terms of Down syndrome, yes, that individual can develop pretty much normal, all right? About 5 to 10% of all fertilized human eggs are going to result in an embryo with an abnormal chromosome number, all right? And about 50% of all spontaneous abortions are due to an alteration in that chromosome number, all right? The body's sensing that something's not right. It's going to spontaneously abort that fetus. And we know that some abnormalities can survive. Any questions on this? All right, I am going to change the slide now. Now, if we're talking about plants, changes in chromosome number is actually pretty common. All right, it is common for plants to exhibit polyploidy. All right, they're often more robust and have a greater adaptability. It lets them be more flexible to the environments that they're in and adapt to new conditions. That can be important in agriculture. Yeah.
I'm just wondering, is like a 4-leaf clover a chromosomal thing or more like a genetic inheritance thing?
Ooh, I do not know. That is something I would have to look up. That's a good question. Yeah. So maybe if some of you have extra time after class day, you can look up. Is a 4-leaf clover a chromosomal abnormality? Right? Because we know in plants, polyploidy is pretty common. All right. So you've already done this homework. We're going to move on to the next slide. This table I presented earlier, and it's up there for your reference. All right, so we're going to look at Mendelian inheritance. All right, so theory of heredity. is that storage of information in DNA is common to all living things. All right, plants have DNA, we have DNA, frogs have DNA. All right, DNA is pretty common between all living things. All right, and DNA is packaged in these discrete units and is passed along to the offspring. We now know that those discrete units are chromosomes. All right, we're passing along the genetic information via chromosomes. Now, if you've learned about genetics in any capacity whatsoever, you have probably talked about Gregor Mendel, right, and his pea plant experiments. We're also going to talk about him because it's just such a monumental experiment and a lot of great things came out of it. All right, so he experimented on pea plants and plant hybridization in 1866, right? But nobody really looked at this too much closely until about the 1900s. All right, so it was actually rediscovered in the 1900s. So you could say he was ahead of his time. All right, but he discovered some important things. All right, so heredity for one, the transmission of traits from their parents to their offspring. All right. As well as what is a trait? Well, that is a characteristic of an individual, right? So maybe you have the trait for brown hair, maybe blonde hair, black hair, so on and so forth. That is a trait. And that is a trait that has the potential to be passed along to your offspring. It is hereditary. All right, so Mendel is asking some pretty basic questions. All right, so he was asking, why do offspring resemble their parents? All right, if we look at a puppy here, why does it look like the mother? All right, and how does this transmission occur? All right, what is the process by which this is happening? So Mendel used the common garden pea, and he used this pea plant because it had some really awesome traits that made it easy to work with. All right, it's really easy to grow. All right, it doesn't need anything special. It has a really short reproductive cycle. All right, so if we're working with a mammal, maybe we'd have to wait a couple years before another reproductive cycle would come, best case scenario. All right, produces a large number of seeds. All right, we can get a lot of offspring that we can look at. The matings are easy to control, and the traits are easily recognizable. All right, so when I say the matings are easy to control, right, we're not getting too much into flower anatomy here, but I wanted to point out how this happens. All right, so if we look at our flowers here, we have a purple flower, we have a white flower. All right, we are going to remove the stamens on the purple flower. So the stamens contain the pollen on this purple flower. We are removing it so that these male components cannot interact with the female component. All right, so we're removing it, that way it cannot self-fertilize. All right, so if you're self-fertilizing, you're using your own pollen to fertilize yourself. All right, so we're removing the stamen so it cannot self-fertilize. All right, so there's no mistaking who is the parents of the offspring. And then we're going to use a paintbrush, take some pollen from this white flower on the white stamens, move it over to the female part of the purple flower, female part being called the carpel. All right, and that carpel is going to develop, going to mature into a pea pod. All right, so it's really easy to finitely control the matings in pea plants. All right, and often we'll see that the offspring are going to be called F1. All right, that stands for first filial, means son or daughter. All right, so first son or daughter generation. All right, so what traits did Mendel study? Pea plants have a lot of easily recognizable traits. Well, he studied 7 different traits. He studied flower color, flower position, so are they on the sides of the plant versus the top, the seed color, the seed shape, are they round, are they wrinkled, pod color, pod shape, again, are they smooth or do they have these constricted segments along the pea pod, and then the height, are they growing tall or are they dwarfed? All right. So he was looking and observing these traits, and the observable features compromise its phenotype. So if I ask you what the phenotype is, it is what you can physically see from an individual. All right. So it just so happens that Mendel's pea population had 2 distinct phenotypes for each of these 7 traits. right? So he could look at a flower and determine the phenotype because he could see one phenotype was purple, one phenotype was white. All right, another key thing that Mendel worked with is Mendel worked with pure lines. All right, there are lines that have the established genotype. All right, we know what they are. When they self-pollinate, they're going to produce identical offspring. All right, sometimes called true breeding. All right, so he used these pure lines to create hybrids. All right, so if we mix two different pure lines together, we're going to get a hybrid in the F1 generation. All right. So Mendel did a couple of different things. The first type of experiment he did was a single factor cross. All right, this is where the experimenter is only following one single trait. So if we look at our image here, what we are looking at throughout this entire experiment is height. All right, we are only looking at height. We don't care about the flowers. We don't care about the color. We're just looking at height. All right, so we're starting with that P generation, the true breeding parents. We are breeding them together to create the F1 generation. All right, and these are called monohybrids. Mono meaning one, all right, hybrid because we've crossed two pure lines, all right, and so we're only looking at one trait in our hybrid cross, all right, monohybrid. Now if we self-fertilize this generation together, we can get the next generation, which would be called F2, all right, and this is where we start to see a recessive trait start to appear. All right, so let's look at why this happens a little bit more. All right, so we have dominant and recessive traits. I'm sure everyone has heard about this in some way, shape, or form. All right, dominant traits are going to be the displayed trait if that trait is present at all. All right, if that trait is present, we are going to see it. A recessive trait is going to be masked by the presence of a dominant trait. All right, it's only going to be displayed if this individual only has a recessive trait. All right, so if it has both a dominant and a recessive trait in its genes, we're only going to see the dominant trait. All right, and we can see that here. All right, we see that the recessive trait is this wrinkled seed here. It only shows up about in a 3-to-1 ratio here. All right, so the dominant phenotype is hiding the expression of the recessive most of the time, but we also get this recessive phenotype. All right, so we now know that the hereditary determinants for a trait are now called genes. All right, so if we look down here at our fruit fly genes, all right, we see that we have genes on these homologous pairs coding for eye color. All right, now Mendel also proposed that each individual has 2 versions of these genes. All right, so coding for the same type of genetic material, but there's variation in this gene. All right, and that variation would be called an allele. All right, so looking back at our gene for eye color, one allele would be for red eyes, one allele would be for purple eyes. All right, and it's these different alleles that are responsible for the variation in the traits that Mendel discovered. All right, so if we had the same gene on both sides, we wouldn't see variation. All right, if it only coded for red, there was no variation, we would only ever see red. All right, but because he saw 2 different versions of whatever he was looking at, maybe it was flower color, right, he determined that we must have 2 different alleles determining what that gene is. All right, so again, I just want to reiterate that the alleles found on an individual is going to be called the genotype. All right, but if we're looking at the effect of the genotype, we're looking at the phenotype. All right, so the phenotype is what we see. The genotype is what underlies what we see. All right, so often it's denoted by this by letters. In this case, we're looking at tall and dwarfed plants. So big T, the dominant allele, is always going to be uppercase. Little t is the recessive allele. It's always going to be lowercase. All right. So this TT here, this is the genotype. All right, genotype TT. Now because it has a dominant allele on it, the phenotype is going to show up as a tall plant. All right, because that is what we are seeing. We are seeing the phenotype. Now the physical location along this chromosome, that is the locus. All right, so we might have a chromosome that has many different genes. If we want to map out where that gene is, right? Say we know that this gene occurs on this specific part of our chromosome. That is that gene's locus. All right, that is a lot of terminology thrown at you. All right, so I want to take a step back, and I want you to fill in the blanks in this chart below. All right, and I want you to use the letter Y to determine the alleles. So give it a shot. Now let's make sure we're all on the same page before we move forward. Must be the first Wednesday of the month.
This literally is the thing in Wisconsin. It's just
Really?
weird that they're doing it. They do it in November. They never even like realized it's not a thing anymore. What? Since when was there ever a tornado?
Yes.
It's a demonstration. The eye thing. Yeah. It's I taught John how to do it.
Bro, you can like talk.
What would like the like big Y little
Yes.
that's from
What is the genotype you're looking at? So in this case, we're looking at pink color. Yep, yep.
So that means that it would be
Could be. This is yellow. The dominant is present.
okay.
Yes. There's another genotype that we will do a couple, and then you'll do it more in lab. All right. Has everyone had a chance to think about this? All right. So first of all, what is a phenotype? In general.
The appearance of the individual.
Yeah, physical appearance is what you are seeing on an individual. All right. What is the genotype?
The alleles received from
The alleles received from the parents that are going to determine what the phenotype is. Yes. All right. So if we're talking about alleles, right, and we know we're going to have two alleles, right, one from mom, one from dad, and we're using the letter Y, what might the genotype be for the dominant yellow key? In terms of Y's, big Y, little y? Yeah, so that could be one, totally. Big Y, little y. We have the dominant big Y that's going to say that we are going to see the yellow. What's another version? Big Y, big Y. Yeah, remember we have two alleles. And so if both of those alleles happen to be dominant, we're also going to see that dominant phenotype. All right, so if we're looking at the recessive genotype, what might that be? Little y. Yes, we cannot have a dominant allele in our genotype because that will mask out the recessive. So they both have to be recessive. All right. So I put the answers up on the next slide here. So when we look at the genotype, we have different words that we can characterize for each of them. All right, so if we're big Y, or little y, We are homozygous. We are an individual that carries identical copies of the same allele for a given gene. All right, we either have both recessive or both dominant. Now we can also be heterozygous. All right, it's going to be an individual that carries two different alleles for a given gene. All right, so we're going to carry the recessive and the dominant. We're going to carry both of them. We are heterozygous. All right, does that make sense? Any questions about genotypes I can clarify? All right. So if we look at a cross between two homozygotes, right, we can draw these Punnett squares. And the Punnett square for a homozygote is pretty easy. All right. So the father only has one allele, big R. So that's the only option we can have. All right. And mother only has one type of allele, little r. That's the only option we can have. Their offspring can only have this combination of big R, little r. All right, in this case, we're looking at the roundness of the seed. All right, and round is the dominant trait, so the seeds are going to appear round. Now, if we look at two heterozygous individuals, we can draw this Punnett squares that have 4 squares to them. It's a 2 by 2 square here. All right, the father has both the dominant and the recessive, so we have two options for the alleles that can be passed on. The same for the mother, all right? We have two options, the dominant or the recessive. All right, now if we combine these together, we can understand what the offspring might have for a genotype for that gene. All right, so we could have big R, we could have little r, little r. We could have two that have big R, little r. All right, so this would be a Punnett square and how we draw them. All right, so if we look at the ratio here, we see that it's going to be a 1 to 2 to 1 ratio of genotypes. All right, 1 RR, 2 big R, little r, 1 small r, small r. Yes? Or is that just a stretch? A stretch, okay, that's fine. All right, we can also look at the ratio of the observed phenotypes. We see 3 of them are going to be round, 1 of them are going to be wrinkled. A 3 to 1 ratio. All right, so I have another exercise for you. All right, looking at the flowers on the right there, I want you to speculate the genotype for the P generation, the F1, and the F2. All right, use the letter P to denote the dominant and the recessive allele. And in this case, we are looking at the color purple. All right, purple being dominant. And think about the ratio of purple to white flowers in the F2 generation.
Second generation is the 25. I need to do it. Am I right though? I don't know why I can do that in my head. I wasn't able to do it. I have a question. No? No? Oh, there it is. 1 of 3. It's okay. I'm in the thread, so. Yeah. No, I want to go back to the I don't know what it is.
Yes, self-fertilized.
Yeah.
right, so let's think about the genotypes of our flowers. We're looking at the P generation. We know it's a true breeding line. All right, what is the genotype for the purple flower? Big P, little p. Yes. What about the white flower? They both have to be lowercase. Yes. All right. So if we're looking at the F1 generation, what might be the genotype for this? Big P and little p. Yes. One has to be dominant because that's the phenotype we're seeing. Right? But we know it was crossed with a genotype that was all recessive. So that recessive is in there. All right, so now let's look at the F2 generation. What is one possible genotype for this flower? Big P, little p, that's one combination. What about for this next flower? Two big P's, yes, big P, big P. What about the next combination? Another big P, little p. Yeah, so those are the three ways that we can get that dominant trait to appear. All right, big P, or big P, little p. Right, now two of these are going to have the big P, little p. What about this white flower? What is the genotype there? Little p, yes. We know they're both little because they're showing the recessive phenotype. All right, so what is the ratio of purple to white flowers in the F2 generation? 3 to 1, yes. We see 3 with the dominant phenotype, 1 with the recessive phenotype. All right, will this always be the outcome when we're looking at this type of cross? Yeah, it will be. Yeah, if we're looking at pure lines, and we do this type of cross where we're getting the F1 generation and self-fertilizing itself, we're always going to see this 3 to 1 ratio. All right, and this is where we get into Mendel's law of segregation. All right, so two alleles of a gene are going to separate from each other, all right, and give rise to gametes so that every gamete receives only one allele. All right, so if we look at that in terms of meiosis here, all right, we can see that the parent here has two alleles. Once we've gone through meiosis, we are giving rise to gametes that only have one allele, all right, because they are now haploid. All right, does this make sense? In terms of meiosis, back a slide. All right, so this is one of Mendel's laws that he came up with, the segregation of alleles. All right, and if we're looking at meiosis, at what point might this happen? What step kind of segregates out these alleles? Anaphase 2. Yeah, anaphase 2 is when we're separating out those sister chromatids. All right, now it'll eventually go through telophase and create its own individual cell, but anaphase 2 is where we are separating them out to only get one allele for each cell. All right, and we can see one allele for each cell. All right, also with the principle of segregation, all right, these F2 traits, this F2 generation, is going to follow approximately the 3 to 1 ratio that we saw of dominant to recessive. All right, this is exactly what Mendel saw. All right, so I put both images up here. They both say the same thing. All right, this one just has the pretty pictures associated with it. But if we look at the ratios here, they're all about a 3 to 1 ratio. All right, so dominant is going to be present in a 3 to 1 ratio. Let's see if I can, there we go. All right, so let's try our hand at Punnett squares for the last few minutes here. Draw a Punnett square with the possible genotypes of the offspring of this mother and father. All right, and F is indicating presence of freckles. And I put this Punnett square up there so you'd have a reference to follow. All right, how are those Punnett squares coming along? I guess the one Punnett.
Okay.
Yes. All right, so we have a couple minutes left in class. Let's go through the Punnett square together. All right, so the mothers. All right, what possible alleles did she pass on? Big F and little f. What about the father?
Big F and little F.
Big F and little f. Yes. All right. So our first square here, combining big F and big F together. All right. What phenotype do we get? Freckles. Yes. All right. Big F and little f. What's the phenotype? Freckles. All right. Same type of cross up here. We know we're going to get freckles. And if we have little f and little f, no freckles. No freckles. So what is the ratio of phenotypes here?
3 to 1.
3 to 1. Yes. 3 are going to show the dominant. 1's going to show the recessive. All right. What about the genotypes? What's the ratio there?
25, 25.
So it'd be like a 25, 50, 25. All right. 25% are going to have big F, 25%, and I put an F there. It's going to be little f and little f. 50% is going to be big F, little f. Right? The easier way to say that is a 1 to 2 to 1 ratio. All right. Let's end here today, and we will finish this lecture on Friday. One quick note, there is a video that I would like you to watch because I'm writing. And if you have that resource assignment, a third of you will have that on Friday. Yeah, there's a quiz.
His official title is like
Yeah. We didn't get to our whole life personal second half.
No! It's like the best way I can describe it. Why? Is your back? Oh, really?
Gone Monday.
Yeah.
I just went through the slides.